All right, let's get started. It's my great pleasure to introduce our speaker this time for the Connect Science. So first of all, there is a link from this page you can see here. Based on this link, you can look up for the upcoming talks, and also all the previous talks are recorded that you can see on Zoom, on the recorded videos. And during Alex's talk, if you have any questions, please post it on the QA section, not in the chat section. And we will be reading out those questions after the talk. So let me introduce Alex Marston, our speaker. So Alex received his MD from Harvard Medical School and PhD from MIT, working with Rick Young and Rudy Yanish. After residency at the Brigham, he has been on the faculty at UCSF since 2012. He's now an associate professor of medicine and is the founding director of the Gladstone UCSF Institute of Genomic Immunology. Dr. Marston's group employs integrative genomic approaches to decode the genetic programs governing T-cell subspecialization. They aim to connect the transcriptional regulatory circuitry of T-cells to human genetic studies and functional genomic screens to advance understanding of specialized T-cell function and potentially generating novel therapeutic strategies for immune-mediated diseases. Dr. Marston is an investigator of the Chan Zuckerberg Institute, the Parker Institute of Cancer Immunotherapy investigator, and also a recipient of the STAR Award of the Cancer Research Institute. He received a career award from Boris Wellcome Foundation and is a new innovator award from NIH. So without further ado, let's welcome Dr. Marston.
Thank you so much for having me and giving me a chance to tell you a little bit about the work that we're doing in the lab here at UCSF and at this new Gladstone UCSF Institute of Genomic Immunology. We have really been focused on trying to use genome engineering technologies, especially CRISPR, to go in and test which sequences in the genome are required for specialized functions in human T cells. And now more and more, we're thinking about how we can use these gene engineering technologies to actually program T cells to behave how we want, to make them more effective as a new type of medicine, where the medicine itself is a genetically engineered cell. See if I can advance my slides. Yep, so before I get into it, here are some financial disclosures that I'm supposed to show you. And this idea that we can move beyond small molecules or biologics into genetically engineered cells as a new way to treat disease is not science fiction. This is now happening. As many of you will know, one of the leading edges of where we're seeing this actually achieved is with CAR T cells. So briefly, CAR T cells are a patient can go in, have their T cells removed from a blood sample and then genetically modified outside of the body in the lab. The way that is currently done for CAR T cells is that a clinical grade virus is used as a vector to insert the genetic material for a chimeric antigen receptor, or CAR. The current viruses that are used are either lentiviruses or retroviruses that insert the transgene into non-targeted sites in the genome, but that's sufficient. The transgene gets expressed, the CAR gets expressed on the surface of the T cells, and then these genetically engineered cells can be reinfused into patients, and the T cells will be directed by this artificial antigen receptor to find a target cell, the cancer cell. And that has been remarkably effective for certain types of cancer. There are now a few different FDA-approved CAR T cells recognizing CD19 leukemia, certain types of CD19 leukemias and lymphomas. These are FDA approved. It's been remarkable. There's still a number of limitations. One of them is the cost. I do want to highlight this, these are still hundreds of thousands of dollars per treatment. There's a number of drivers of this. One of them is the cost of the viral vector. but broadly manufacturing these still remains a challenge. And second is that although this is effective for certain types of cancer, this still has not been generalized to most types of cancer, especially solid tumors, which are major causes of cancer mortality. So we have really thought that what we're seeing is just the tip of the iceberg of what's possible with adoptive cell therapies. Broadly, we'd like to expand the power of genetically engineered cells to treat human diseases. And we think that that is enabled by the major advances in our ability to engineer the genome with nucleases, especially CRISPR. And what we'd like to be able to do is, in the context of cell therapy, to be able to take human immune cells and remove genes that are limiting the function of those cells, fix individual mutations from patients to improve the function of immune cells. Or what's been technically the hardest, but where we think there's enormous possibilities, is to take a big stretch of DNA and either insert it not at a haphazard site the way that viral vectors do, but at a targeted site in the genome, or replace a large stretch of genome sequence to really, in a targeted way, reprogram that aspect of the DNA code. So let me just briefly tell you what I'm going to talk about today. I'm going to divide the talk into 3 sections. I just very briefly, I'm going to give you an update on our ability to do these types of genetic manipulations in human T cells and tell you where things stand. But then I really want to talk about, as we've gotten better and better at genetic engineering and moving that towards therapeutic applications, the next big question that we realize is, what are the genetic edits that we want to make to T cells to have the biggest and most powerful effect on improving their function in treating disease? So I really want to talk today about 2 different discovery platforms that we've set up in primary cells in recent years. One is the ability to now do genome-wide discovery of which genes can be manipulated to tune the function of T cells. And the other is more recent work that we're just really expanding now, which is not genome-wide knockout screens, but complementing that with high throughput pooled knock-ins, where we knock in lots of sequences into the same site and see which one has the biggest effect on the cell phenotypes that we care about. So with that, let me just show you a little bit about what we've done over the past couple of years. This summarizes a lot of work, but briefly, we've set up a platform that we think is very easy to use, and we've shared it now with a number of labs. We can purify human immune cells from blood samples. So these are primary human cells. We had a lot of problem figuring out how to get Cas9 and guide RNA efficiently into those cells, especially the Cas9. And the turning point was really a collaboration with Jennifer Doudna's lab, where we started using recombinant Cas9 protein as the nuclease rather than a nucleotide encoding that Cas9. So we use recombinant purified Cas9 protein and load that in vitro with a guide RNA. Those guide RNAs are now available from a number of commercial vendors. They're chemically synthesized. You can design them and have them in about a week. Cas9, we have big stocks of the protein that we grow in a core here. We put them together to make a ribonuclear protein, or RNP. We found that RNP can then be electroporated into human T cells and very efficiently disrupt a target gene. And within a matter of days, we can go test the consequences of that genetically edited cell. The other thing that we've realized is that it's not just useful for knocking out genes, but we can actually knock in new genetic sequences using the cell's homology-directed repair machinery. So we can actually co-electroporate a naked piece of DNA, again, this is non-viral, with homology arms to either site of the CRISPR cut site, and use that template to knock in a targeted set of nucleotide replacements. And we've gotten these bigger and bigger over the past couple of years. I'll show you some of what we're able to do with that. But we can electroporate in the DNA template, and we can actually start seeing knock-in DNA edits and their consequences within about 2 weeks. I'll just show you, I'm not going to talk about any of the data, but our recent paper in Nature Biotech at the end of last year made some technical improvements to this that not only increased the efficiency, but now expanded the set of cells that we can manipulate with this CRISPR-based non-viral platform. We can now do this not only in bulk T cells, but we can do it in subsets, CD4, CD8s, regulatory T cells, gamma delta T cells. It also works in B cells, NK cells, and CD34 positive cells. So we think this may also be a way of going after the hematopoietic stem cell population. So we think that any cell that we can get to culture and proliferate well in vitro could potentially be amenable to this platform. So where are we going to actually use this power to go in and do targeted nucleotide replacement? I want to talk briefly about where we're planning our first clinical trial, we hope. We're working towards this very actively. This is for a family that came to our attention. This is a family that has 5 children, 3 of whom are affected by different manifestations of autoimmune disease. One child developed type 1 diabetes. Another sibling doesn't have type 1 diabetes yet, but has multiple autoantibodies suggesting a very high risk of type 1 diabetes. And a third, an older sibling, actually doesn't have type 1 diabetes, but has very severe constellation of autoimmune attack with including autoimmune neutropenia, ITP, and has required immunosuppression with steroids on and off, and has been sick enough that the family has actually been advised to consider the possibility of getting a bone marrow transplant for this child. The family has been resistant, knowing some of the risks that go along with bone marrow transplants, and has been looking for alternatives. And we've been trying to see if there might be an alternative. Now, because this family has 5 children, 3 of whom are affected, it raised the possibility that this family may have a Mendelian cause of autoimmune disease. So the family underwent exome sequencing, and the mutations are actually very clear. Each parent, by bad luck, has a different mutation in IL-2RA, the high affinity receptor for IL-2. And the 3 children that I told you about with different manifestations of autoimmunity are the ones who, by bad luck, inherited both mutant copies. IL-2RA has a number of roles in the immune system, but one of the places where it has a critical role is in regulatory T cells, the cells that suppress autoimmunity, suppress inflammation. And it turns out that these children have cells that kind of look like regulatory T cells, but they don't work properly without IL-2RA on their surface. So these children had seen specialists around the country at Yale and Stanford and other places. And actually, Kevin Harold called us up and wondered whether CRISPR may be an opportunity to intervene and correct the regulatory T cell-like cells from these children. So as a step towards that, we actually have gotten blood from these children mailed to us in the lab. And you can see that on the flow cytometry here from a healthy donor, if we stimulate, these are stimulated T cells, most of the cells will turn on IL-2RA on their surface. Now, as a result of the mutations, these children's cells really fail to adequately induce IL-2RA on their surface. But we were able to actually go in and pick one of the 2 mutations and do a targeted correction with a small homology-directed repair template and restore meaningful levels of IL-2RA to their surface. Now, we've continued to work on this, but we really benefit here from UCSF's long investment in regulatory T cell therapies to treat autoimmunity and for organ transplants. This is work that has been set up over a number of years by Jeff Bluestone and carried forward by Kizhi Tong and now by Jonathan Essenstein, who's the clinical head of this cell therapy division for regulatory T cells. That has been really taking patients' regulatory T cells, sorting them based on a few markers, expanding them ex vitro, try to get more regulatory T cells, and then reinfusing them for a number of clinical trials. What we have tried to do now is to insert into this clinical grade manufacturing process the gene correction step. And Brian Shai is actually the first cell therapy fellow at UCSF, who was jointly advised by me and Jonathan Essenstein, and he's going through all the steps of manufacturing. We've had, usually regulatory T cells get sorted initially based on their IL-2RA. We've not been able to do that. We've had to work at a different panel of flow markers to enrich for the FOXP3 positive demethylated Tregs, do the gene correction, and now expand them and go through all the safety testing. We're working very closely with the Innovative Genomics Institute, which was founded by Jennifer Doudna as a Berkeley UCSF partnership. And I'm working closely with my colleague Theodore Ernov there. And then these patients are being seen by Kevin Harold. So this is very actively under development, and we hope to have a clinical grade gene corrected regulatory product to infuse into one of these children, hopefully by the end of next year. we hope that this helps this individual, and we also hope that this creates a general approach to be able to pick cells in the immune system that are not working properly as a result of mutation and go and fix the underlying problem. And we hope that CRISPR is sufficiently flexible that this will be something that allows us to go after Mendelian diseases of the immune system broadly, whether it's targeting regulatory T cells for some patients or targeting other T cell populations or even hematopoietic stem cells. So this is an expanding effort here in the Bay Area. That's an example where there is one mutation that we want to actually go in and correct. But for many therapeutic applications, especially cancer, one of the things we need to do is be able to more broadly reprogram immune cells to enhance their function for treating cancer. And one of the programs that we care the most about is what the T cells will recognize. T cells naturally have a polyclonal repertoire of T cell receptors, and that's important to protect us against the diversity of potential pathogens we'll encounter. But for a therapeutic product, we'd like to define the antigen specificity of T cells, whether it's by putting in a defined T cell receptor or by putting in a chimeric antigen receptor. Now, we wondered whether this non-viral platform could do that. And we wanted to test this by seeing if we could actually go in and replace the cell's own T cell receptor. The trick here is that a T cell receptor has two components. It has a TCR alpha and a TCR beta, T cell receptor alpha and beta. And we have to, in order to get the antigen specificity, we have to replace both simultaneously. So through a collaboration with Tony Rebus's lab at UCLA, we came up with a strategy to do this. The idea was to make a single cut in the constant region of the TCR alpha region, in the constant region, and then to use homology-directed repair to actually insert a polycistronic sequence that encodes the full-length TCR beta and TCR alpha. We realized we actually don't have to put the full-length TCR alpha in. We only have to put in the variable region because we're actually going to fuse it in frame with the constant region. So that actually allowed us to compact the sequence a little bit. And we separate the TCR alpha and beta with a peptide excision sequence of P2A. We wanted to see if this would work in principle. So we started with a very well-characterized TCR that recognizes the NY-ESO cancer antigen. So you'll see here, this is inserting 1,500 base pairs roughly of sequence, which was, at the time, we were as a technical challenge, but this worked beautifully. And you can see compared to the control, what I'm showing on the vertical axis is dextramer staining for the NY-ESO antigen. You can see in the control population, you don't see T cells that recognize this antigen. But with this non-viral cut and paste maneuver, we actually create a relatively large fraction of cells that are now specific for this cancer antigen. You can see that compared to the controls, in many cases, we're actually knocking out the TCR. And of the cell, that's through non-homologous end joining, presumably at the cut site. But of the cells that remain TCR positive, actually about 2/3 of them now have been programmed to have the antigen specificity that we want. And we put these cells through a whole battery of functional tests. In vitro, they will recognize cancer cells based on the NY-ESO antigen. They will secrete the right cytokines, they'll kill those cells. It's also MHC restricted. And importantly, in vivo, these human cells, if you put NY-ESO positive cancer cells into an immune deficient mouse, the non-virally engineered TCR replace cells will home to the site of the tumor and control tumor volumes. So we are really pushing also on this as a general platform, not only for this TCR, but broadly to replace TCR repertoires, and also for insertion of a targeted chimeric antigen receptor into a specific site of the genome. And all of this, again, has the benefit of being done without the requirement of viral vectors. So that's the platform. In the remaining time, I really want to talk about how can we make these cells behave even better? We know that if we're going to go after solid tumors, we need to somehow not only make a T cell that will recognize the cancer, but have boosted efficacy in actually clearing that tumor once it finds it. And here we've been inspired by what other groups have done in cell lines and in animal models with really doing forward genetics through genome scale CRISPR knockout screens. And we wanted to see if this was possible directly in the primary human cell that would be a therapeutic product. And what we found was that we could get the guide RNA libraries to be transduced with lentivirus into T cells. We've had more problem with transduction with the Cas9. So what Eric Schiffert, and who's a postdoc in my lab, and Julia Carnevale, who's an oncologist and a postdoc in Alan Ashworth's lab, they teamed up to come up with a strategy to overcome this challenge. What they do is they put the guide RNA and library in with lentivirus, and then they electroporate the cells with Cas9. So Cas9 will go in as a protein. It'll find the guide RNA that's expressed off the lentiviral integration site. And although the protein will only presumably stick around for a short period of time, it's there long enough to make a permanent genetic edit to those cells. And on top of that, those cells will also not only be edited, but they'll be barcoded with which edit they get based on the lentiviral cassette that encodes the guide RNA. So we end up with a population of cells from an individual human, where each cell is has a different gene disrupted, and that gene disruption is barcoded with the lentiviral integration cassette. And then we can subject these primary human cells that have been edited to any phenotypic screen or selection. So we first just wanted to say, could we find all the genes in the genome that determine the extent to which T cells will proliferate when they're re-stimulated? We wanted to see, could we find cells that, and the way we did this was we CFSE labeled the cells after the gene editing. We re-stimulated them, and we did a big FACS sort. Eric and Julia did a big FACS sort to separate the dividing cells that have low CFSE from the non-dividing cells that have high CFSE. We asked which guides drive which phenotype. What we found was that we could immediately take a off-the-shelf library from the Broad with 80,000 guide RNAs targeting roughly 20,000 genes and start systematically discovering genes. There were a large number of genes that when we knock them out, make the cells proliferate less well. Those are shown here in red, and those are not random. If you look, they actually neatly recapitulate all the biochemistry that's gone in over many, many years into decoding the T cell receptor signaling pathway. And essentially every gene downstream of the TCR, or many, are shown here in red, saying that indeed the genetics confirm that these are essential for the normal response to restimulation through the TCR pathway. So that was really nice confirmation that we're finding real biology. But even more exciting to us were the genes shown in blue. And these are genes that when you knock them out, make the cells divide more in response to restimulation. So we think that these potentially could be very interesting targets for cancer immunotherapy to boost the proliferative capacity of T cells. So we wanted to follow up on these a bit more. And we took advantage of, I showed you the pooled screen. We've also put a lot of effort into setting up a high throughput arrayed screen. So we have a 96 well plate electroporator where each well can electroporate primary cells with a different RNP targeting a different gene. So we took hits from our screen and wanted to confirm, first of all, the effects on proliferation. We wanted to see if they affect other markers of activation. So we could actually start doing multi-phenotyping in each well. And the other thing that doing in this arrayed format does is allowed us to look for a non-cell intrinsic phenotype. We could actually engineer the T cell and then see, does that boost the ability of those T cells to actually kill cancer cells? So briefly what this looks like is we picked a couple of top hits from our screen, knocked them out, CFSE labeled them, and the effects of CFSE nicely confirm what we saw. So these were some of the top hits that boost proliferation. You can see CFSE dilutes more as a response to these knockouts in multiple human donors and with multiple guide RNAs. Here's a gene that makes the cells divide less well, and you can again see that the cells retain CFSE high. The other thing that was nice about this is it allows us to confirm, at least for these genes shown here, that it depends on the stimulation. You don't see this if in the unstimulated population, meaning that these gene knockouts are not causing unregulated proliferation. It's proliferation that depends on stimulation. So that'll be important for finding good targets for cancer immunotherapy. And this wasn't limited to just these two relatively well-characterized genes. Actually, most of our hits could be reconfirmed this way, suggesting that this was really finding bona fide hits to boost proliferative capacity of T cells. We not only looked at proliferation, but some of the other markers that characterize how T cells get stimulated, CD8 T cells get stimulated, including we looked at CD154 and CD69. And you could see that things that made the cells proliferate better tended to also boost levels of CD154 and CD69 compared to controls. So this seems that we were really enhancing these T cell stimulation response. And then we wanted to see, this was not what we screened for, but we wanted to see if it was actually something that we could discover through this screen. Did this boost the cancer killing capacity of these cells? So here we made antigen-specific T cells. Here we do it by inserting the TCR with a lentivirus to recognize the NY-ESO antigen. And we use the RNP to just knock out the candidate genes. So we end up with antigen-specific cells with different genes knocked out in each well. And then we co-culture them with NY-ESO positive cancer cells, the A375 cells, that are labeled in red with RFP. And we could actually just visually see, do any of these individual gene knockouts boost the ability of T cells to kill the red cancer cells? You can see we actually found 4 genes here that when we knock them out in T cells, make the T cells better at clearing the tumor compared to controls. And we could actually watch that. We use the IncuCyte device with live cell imaging. You can actually see the kinetics that these 4 genes actually make the T cells better at killing cancer. So we're really excited about this as a platform, also finding individual candidate genes that could be developed either as drug targets or even more, potentially more promisingly, given these genes, as targets for gene modification in cell therapies. This is unpublished data. I want to give you a little bit of a preview of where we're going. We have been really interested in these genes, and you can see that the gene here that's actually has the biggest effect on cancer cell clearance is RASA2. So we've done a lot of work of looking at what the effects of RASA2 are in T cells. I'm not going to show all of it, it's unpublished, and we're continuing to work on this, but I'll just say RASA2 seems to really boost T cells' response and resistance to a lot of the things that will normally limit T cell function. One of the things we care a lot about for T cell going after solid tumors is their ability to continue to have function even through repetitive exposure to antigen. Many T cells will lose their function, either become exhausted or dysfunctional, as if they chronically are exposed to antigen. So we've tried to mimic this in vitro by repeatedly adding the A375 cancer cell line with the NY-ESO antigen multiple times over many days. And I'll just show after the first stem that we can again recapitulate that the RASA2 cells do a better job of clearing the cancer cells than their controls. But the effect becomes even more pronounced through repetitive stems. So we're really excited about this as something as we'd like to program T cells to do. You can see that they continue to be good at cancer clearance even after these late stimulations, repetitive stems, unlike their control counterparts that will over time start to lose their cancer killing capacity. So that just gives you a little preview of how we're really thinking about what are the phenotypes that we want T cells to have, how can we discover genes and then start testing and confirming that they've actually enhanced those phenotypes that will have benefits for treating disease. So as we start to find genes that are interesting based on their phenotypes, we'd also like to be able to deeply characterize them. And one of the tools that's emerged in the field as a powerful way of characterizing the effects of any gene or any perturbation broadly is single-cell RNA-seq. A number of groups have developed different versions of coupling CRISPR screens, pooled CRISPR screens with single-cell RNA-seq called Perturb-seq or Crop-seq. Roughly, the idea is to do single-cell sequencing with a droplet-based platform to end up with single-cell transcriptomes from a population of cells that have undergone different genetic perturbations in each cell, and to capture the transcriptome of each cell, but also capture a barcode that tells you which gene was knocked out in each cell, so that you can actually start pairing genetic perturbations with the consequences on the overall transcriptome of a single cell. We realized that we could actually do this now in primary cells. So we took a number of hits from our screen, as well as some genes, guide RNAs targeting well-characterized checkpoint genes, and made a targeted library in the crop-seq vector, infected the cells with crop-seq vector, and then electroporated them with Cas9 protein. We did this from 2 human blood donors, D1 and D2, and we did it both in the resting cells after the genetic perturbation, and then in cells that get re-stimulated. We subjected all these cells to the single-cell RNA sequencing. And what I'm showing you here first is just a dimension reductionality map showing the overall map of cell states that we were able to capture across all the cells in this experiment. And you can see that on this map, the left side seems to be the states that are relatively unstimulated. The right are the stimulated cells. There's some donor effects. But the key question was, where do the guide RNAs shift the state of these cells? So the first thing I'll point you to is the control cells here on the right. The cells that have the control guide are kind of evenly distributed among all the cells. So that's kind of what you'd expect. But we could actually see these marked shifts in distribution based on individual guides. If you knock out LCP2, which is required for stimulation according to our screen, you shift the cells towards the state of unstimulated, more likely unstimulated. These genes that seem to make the cells proliferate better shifted the cells towards the right, towards a more stimulated cell state. We could actually go in and start to look at differential expression by looking at the pseudo-bulked differential expression of different genes in different cells that have different individual gene knockouts, to start piecing together both the shared effects of multiple genes that boost stimulation and some individual effects of specific gene perturbations. And based on these specific perturbations, we're actually able to do correlation mapping to look at the overall effects of different gene perturbations and start finding some perturbations that boosted proliferation seem quite similar, whereas others were potentially more distinct. And this, we think, will be very useful in starting to put together insights into the pathways that we're hitting, potentially to guide us towards combinations of perturbations, either through combined gene knockouts or combining different gene knockouts with immunotherapies. So we think that this is just the beginning of what's possible with bringing single-cell RNA sequencing into these platforms. I'll just say very briefly that, and this is important, that even if we can make cells that proliferate better, that's really only part of the battle. I touched on this idea that the cells not only have to proliferate better in response to their first stimulation, they have to continue to do it if they're going to be effective at treating a solid tumor. The cell will have to clear many cells, so repeated antigen exposure needs to not dull the function of these engineered cells. We have to figure out ways to program them not to get exhausted. But we also have to figure out how to make them avoid many of the immunosuppressive characteristics of a solid tumor microenvironment. That includes, how can we make them resistant to checkpoints? How can we make them resistant to suppressive cells like Tregs that they might encounter inside certain tumors? And how can we make them resistant to suppressive secreted factors like metabolites like adenosine and TGF-beta? So as a proof of concept, Eric and Julia redid this whole genome-wide screen, but now they did it in the presence and absence of an adenosine receptor agonist. They want to see, could we find new genes that would make the cells resistant to this adenosine suppression? And the results were very clear. There are basically two genes that came off the curve that make the cells strongly selectively resistant to adenosine suppression. One of them is an adenosine receptor itself, so adenosine receptor 2A. And it turns out that this agonist that we were using is selective for adenosine receptor 2A. And the effect genetically was limited to that and not to the other adenosine receptors. So this really showed us that the biology was very clean. And then we also discovered a new gene, FAM105A, that's never been implicated in adenosine signaling. And now we are able to firmly say that FAM105A seems to really make the cells resistant to adenosine suppression, and potentially makes FAM105A an interesting target either as a drug development candidate or to knock out to make cells resistant to adenosine for certain types of cancer immunotherapy. So that is an interesting target, but broadly shows that this platform now can be adapted to screen for the effects in multiple different contexts where we want T cells to be effective for treating cancer. Now, everything I've told you so far in this discovery is about the ability to use genome-wide knockouts and to discover genes that, if you disrupt their function, might enhance T cell functions. And that's been very fruitful for us, but we realize it's really only a small sliver of what's possible. And much more can be programmed into a T cell by thinking about, can we discover what gene additions will have the biggest effect on T cell functions? And I do want to give you a little flavor of how we're using that to address the same problem. Can we design T cells that perform better in the tumor microenvironment by adding gene programs rather than knocking out genes? So this is work that has been led and pioneered by Theo Roth, who is an MD PhD student in my lab. And Theo played a leading role in developing our non-viral knock-in platform. And as he got better and better at it, he wondered whether, in addition to putting in just, for example, the TCR beta and alpha, could he insert with that an extra transgene on the same polycistronic cassette that could potentially enhance the function? So you'll see this was a bigger insertion, but Theo's been getting better at this, and he wanted to see could he put in this big transgene at high efficiency. So just as a first proof of concept, he did this with a inert receptor. This is a truncated nerve growth factor receptor and TNGFR. Just wanted to see if it was expressed. And what he found was that this worked again very, very nicely. The dextromer positive cells that recognize the NY-ESO antigen based on the TCR that we put in, almost all of them, if you look with flow cytometry, also express the TNGFR receptor. So we're able to really insert a cassette that has these 3 genes with multiple peptide excision sequences. So this raised the prospect that we could think about what gene would be most beneficial in that TNGFR position instead of TNGFR in that position. And we had a number of guesses, but we wanted to do this systematically. And so Theo came up with an idea to do this as a pooled screen. And the idea was, could we make a single cut in the genome? And now, rather than the screen being different cuts, could we make barcodes on each of the knock-in templates, end up with a population of genetically edited cells where each cell has a different knock-in cassette and a different barcode that tells us which knock-in, so that we could then take this population of cells and subject them to in vitro or in vivo screens, or potentially even single-cell RNA sequencing phenotyping. So this is what the molecular biology actually looks like. For each gene, we put a barcode into synonymous sequence inside the TCR sequence. And we came up with a PCR amplification strategy that allowed us to selectively amplify this barcode only if this template was integrated through homology-directed repair into the genome, or selectively in that case. And we could track the cells that have each of these barcodes in a population. And just as a proof of concept, to make us convince ourselves that this works, we just did a 50-50 mix of cells that were GFP with a corresponding GFP DNA barcode, or RFP with an RFP barcode. And you could see that the protein phenotypes match up nicely with the DNA barcodes. The 50-50 mix can be reconfirmed with the DNA barcodes. But if we just look at the GFP positive cells, they selectively have the GFP barcode, and conversely, the RFP cells have the RFP barcode. So, and we could recover these either from genomic DNA or from cDNA to see which barcode was integrated in each cell. So now that the molecular biology was working, we wanted to design a pilot library to see, can this actually teach us something about biology? Does it actually work? So Theo designed a library of 36 different knock-in constructs. This included some controls, a number of genes that we thought if they were constitutively expressed in the TCR locus from the TCR gene regulatory element, might boost T cell function, including receptors and transcription factors. And other metabolic genes. But most of the genes, what we actually did was we started engineering receptors to make so-called switch receptors. We started taking the extracellular domain of inhibitory receptors, genes, proteins that would normally inhibit the function of the cell when they find an inhibitory ligand. We started making them into switch receptors with the extracellular inhibitory domain coupled now artificially to intracellular activating domain, so that if a cell encounters an inhibitory ligand that it might find in the microenvironment, the cell actually should get more activated. We wanted to see, does this actually work, and which ones are the best? So we took this population of cells with all of the constructs knocked in, and we put them in a number of different constructs and started to ask which barcodes became more abundant, had higher fitness in different contexts. If you just grow the cells in IL-2 and don't stimulate them, we were pleased to see the IL-2 receptor had the biggest effect on the fitness of these cells. Once you stimulate them, it turns out that engineering the fast ligand apoptotic receptor had a big effect in vitro, either by making a dominant negative that was recently published by Chris Klebanoff and Nick Restifo, or by a new one that we designed, a fast ligand receptor with a 4-1BB activated intracellular domain. If you don't co-stimulate these cells, putting in these switch receptors that have CD28 as their intracellular domain seem to have a selective advantage. And if you add TGF-beta to suppress these cells, all of a sudden these engineered receptors that start blocking the suppressive function of TGF-beta and potentially even converting it to an activating signal have a selective advantage. So this really started to show us that in different contexts we could find different genes that to boost the fitness of T cells. We don't only care about this in vitro, we wanted to see could this also work in vivo. And so we actually took a population of cells that are all NY-ESO antigen specific based on the TCR we inserted, but they each have a different transgene in the middle position, and took this input population and infused it into a mouse, an immune deficient mouse that has NY-ESO positive cancer cells in it. Then we asked which barcodes grow out when you compare the input population to the cells that we sort out of the tumor environment itself. And we could see that we actually found, a few receptors that selectively lead to accumulation inside the tumor microenvironment. Either these engineered TGF-beta receptors or actually this transcription factor TCF7. So we were interested to see what is really the effect of these TGF-beta engineered receptors or the TCF gene. Are they leading to accumulation in a way that will also make them better at clearing the tumor, or are they just accumulating in an ineffective state? And to start to answer that, we again turned to single cell RNA sequencing. We wanted to see, could single cell RNA sequencing tell us not only what cells were accumulating, but the state that they're in as a result of the knock-in construct that we put into that particular cell. So we made this is our new version that's analogous to Perturb-seq. We called it PokeSeq. It's pooled knock-in with single cell RNA sequencing. And basically the idea was to make this pooled knock-in cell library, subject them to droplet-based, we use 10x sequencing, and to simultaneously recover the transcriptome plus the knock-in barcode. So again, this worked nicely. Here I'm just showing you that if we do this with GFP and RFP, the GFP positive cells, we can recover with this new molecular biology, we can recover the GFP barcode and the GFP protein level cells positive, and likewise RFP. So this was encouraging, again, that this was telling us the right thing. So now we do a similar experiment in vitro. We took cells from 2 human donors. Now we made the pooled knock-in, and we stimulated all the cells now, but either stimulated them alone or stimulated in the presence of TGF-beta. And you can see that TGF-beta tended to suppress these cells up in the dimension reductionality map up in this upper left quadrant. And we wanted to see what was the effect of all the knock-in constructs. The controls were suppressed in the presence of TGF-beta, and that made sense. That was true for other receptors. For example, this FAST41BB fusion protein also remains suppressed. But now if we engineer the TGF-beta, those cells that have engineered TGF-beta receptors started to show differences in their phenotypes. The dominant negative cells started to escape suppression, and we could even see with this new switch receptor cells that seem to be even more potentially activated as a result, or comparably activated as a result of this switch receptor that they received. And we could actually go in and see that there's cell clusters that these cells were moving into that were characterized by markers that we associate with gene activation. So we think that we're able to now characterize, including by looking at individual genes, the effects of different knock-in constructs. I'll just show you quickly here. The TGF-beta knock-in constructs really cluster together, but only in the presence of TGF-beta. So we can really see a shared effect that's dependent on that exposure to TGF-beta. In the remaining few minutes, I just want to turn back to actually doing single cell RNA sequencing and functional testing in vivo. So we looked at single cell RNA sequencing of the ex vivo population that we used as the input for the treatment, and at the cells that we sorted out of the actual tumor microenvironment. And you can see that there's actually, as you'd expect, a pretty big difference between the ex vivo cells and the cells that have actually made it into the tumor. But even within these cells that make it into the tumor, there's heterogeneity. they tend to all turn on, for example, granzyme, but only a subset of them are turning on interferon gamma. We know that interferon gamma and other molecules can be important for driving an effective anti-tumor response. So we wanted to ask two questions. We wanted to see, with single-cell RNA sequencing, do we find the same constructs that are leading to higher abundance in the tumor-infiltrating population? Are the same ones driving accumulation? And two, which of those that are driving accumulation are also actually driving accumulation in a state that's compatible with clearing, for example, characterized by high interferon gamma? And so what we saw was that when we look at the single-cell RNA sequencing, the correlation of the hits was quite high with if we just looked at the abundance of the barcodes in this independent screen. And the same things come out at the top hit, these engineered TGF-beta receptors and the TCF7 construct. Now, there was a marked difference is that if you look in the TGF-beta constructs, were especially this 401BB, were driving the cells to accumulate in a state where they had high IL-2 and interferon gamma production, whereas the TCF7 tended to keep the cells accumulating, but in a more naive population, they characterized by, for example, CCR7. And that's consistent with TCF7 is a transcription factor that's known to restrain the terminal differentiation of T cells, drive them towards a memory phenotype. So it seems that TCF7 was causing the cells to accumulate, but maybe not going into an effect into the final effector function, which may be important for tumor clearance. We can actually see that if we look at the pseudo-bulked RNA sequence differential expression. For example, interferon gamma seem to be strongly differentially expressed in either the TGF-beta in the TGF-beta 41BB cells compared to the ones that have TCF7 or the control. So the final experiment that I'll just show you data on is we wanted to actually test, is this true? Does TGF-beta 41BB actually lead the cells to accumulate in a state that's going to be beneficial for tumor clearance? And is that not true for the TCF7? And that is what we've seen. The TGF-beta 41BB construct seems to really promote a marked difference in tumor clearance. And the TCF7 did not compared to the control cells. And we think that this difference in the genes in the cell state helps to explain this phenotypic difference that we're seeing with these two different gene knock-in constructs. I'll just show you for the TGF-beta 41BB, the effects are even more marked when you look at all the individual mice treated. The control cells, you can see basically at this dose did not clear these NY-ESO positive tumors implanted in mice. But if we put in the TGF-beta 41BB construct, now many of these mice were able to clear their tumors with these engineered T cells. So we think that this is potentially a promising construct. There may be safety concerns that we still have to look into, but this is something that potentially could advance towards consideration for clinical trials. But even more importantly, what I really want you to take away is that I think we now have a very powerful suite of technologies to start really systematically understanding gene programs that control T cells. We can systematically use gene knockouts to identify sites throughout the genome that are critical for the cell functions that we care about. And then we can use pooled knock-ins at defined sites to systematically test which gene insertions at those sites are most powerful for really enhancing the functions that we care about. And that can be either through replacing the genes with other genes, or actually putting in new synthetic programs at targeted sites in the genome. And we think as we advance with this approach, we really now have an accelerated pipeline to understand the genetic control of cells in the immune system, but also start reprogramming them to drive the generation of new next generation cell therapies for autoimmunity and infections, but heavily for cancer immunotherapy. And we are thinking a lot about how to drive this forward. And with that, I will stop. I want to thank the people in my lab who've done this work and really highlight that this has come out of a wonderful series of collaborations with groups around the world, and also gratitude for our funding sources. And thank you again for inviting me today.
Right, this is a really exciting talk. So we have some questions. The first one is, what is the efficiency of the non-viral transfection, and do you have to sort the transfection positive cells?
It's not a completely easy answer. For knockouts, if we want to disrupt a gene, in many cases we're seeing greater than 90% knockout. We can, if we test a few guides and a little bit of optimization, we can get very, very high efficient gene knockout. For the knock-in, the efficiency is more variable. It continues to improve. One big variable is the size of the construct. The bigger the knock-in construct, the lower the efficiency. I showed you a little flavor. I think we're continuing to improve and continuing to drive that up. But some efficiencies for different knock-in genes might range from 5% to 40%, depending on what we're manipulating. And then the question about sorting, it depends a little bit what you want to do. If we're going to put them in to treat a cancer, for example, it might not be that critical, because the cells that don't have the TCR or CAR, for example, might just be inert, and it's really the dominant function of the cells that have the gene knock-in that you care about. But there may be ways to quickly enrich for those and for certain applications. For example, if we put in a cell surface receptor, we can either sort for those cells or use a bead enrichment strategy.
So do you still, in this case, use the same MOA that people normally use for cancer cell CRISPR screens?
For the knockout screens?
Yeah.
Yeah, the short answer is yes. The same principles apply, that we're tracking the cells based on the lentiviral insert. And if you want to be confident that a gene manipulation is due to a single knockout, low MOI helps you be sure that most cells will have 0 or 1 lentiviral insertions. I always wonder how critical this is. We actually had recently did a different screen in mouse cells with a higher MOI, and actually we're able to still confirm most of our hits. I think you can probably make up for some of that with coverage. I think some of that if you do more cells, the likelihood of any individual combination coming through, even if you get into higher MOI, might be less important. So I think that there's probably some modeling around that, of figuring out what the optimal MOI is for relative to the number of cells that you're perturbing. But the same basic principles apply regardless of whether you're doing this in stable Cas9 cells or electroporating cells with Cas9 protein.
Okay, so we have another question. Surprised you didn't see anything in the calcineurin NFAT pathway.
Trying to think if I have a good answer to that. And I just actually don't remember if we did see things in that pathway or not. I agree that that's a pathway that's important in response to stimulation. Whether that's something that's dominant and is selectively required for the proliferation phenotype that we're screening on, I'm not sure if maybe there's redundancy, but it's also possible. I want to go through our results a little bit more and make sure that I suspect that there are things in that pathway that can make us hits. I just don't know off the top of my head.
Okay. So by deploying the switch receptor regimen, will the mice develop simultaneous autoimmune phenotype?
Yeah, so that's a really interesting question, and something that I think a lot about. So here we are, we're making the cells supercharged. We're trying to make them resistant to all of the things that the body normally does to prevent autoimmunity. We're trying to make the cells that are resistant, right? Like now the cells. So that's one of the, whether this will be perfect or not, we'll have to see. There's ways that I can imagine this being imperfect, but it's one of the reasons I've put so much emphasis into these polygenic cassettes that encode both antigen receptor and these switch receptors. And the idea is that hopefully most of the cells that are getting the switch receptors and are becoming resistant to immunosuppression have also been redirected in their antigen specificity against a cancer selective antigen. So now we're knocking out the cell's own TCR because we're inserting this into the TCR locus. So the cells should be not able to recognize, if they are potentially recognizing a self antigen, hopefully we've gotten rid of that by knocking out the TCR alpha. Plus we put in a new antigen specificity, in this case with a TCR, but it could potentially be with a CAR. So hopefully only those supercharged cells will have a really strong selective bias towards going after cancer antigens and not after other self antigens.
Right. Next one is for the knock-in experiments. What do you do on the regulatory element to ensure the target genes are highly expressed?
Yeah, so it's really interesting actually, and something that we've played with a little bit. But everything I showed you was knocking into the TCR locus, and we know a lot about the TCR is expressed off this TCR's own regulatory elements. So we're not putting in any extra transgene, any promoter or anything there. You could potentially put in a promoter to ensure that it's expressed, and we've played with that a bit. Or you could actually knock in your construct into different genes that have different endogenous gene regulatory programs. And for example, actually, Michel Sadelain did a little bit of this. He used a different knock-in construct. He used a AAV template instead of a non-viral template. But he did knock in CARs into different loci, including the TCR and others, and showed that putting the CAR into different genetic loci does seem to affect its function.
Next one, for the non-viral delivery system, besides like not using lenti, have you tested COSMID or PhageMID, and also what's the cost for this CRISPR RNP system?
It's a good question. We're continuing to test different vector systems, and as we move this towards clinical trials, there is considerable current cost of moving these into good manufacturing GMP reagents or clinical grade reagents. So there is still considerable cost. We do hope that in the long run, these costs will continue to come down as we engineer cells that will be able to be effective at lower doses, as these become more automated and standardized, as more competition enters into the field. So it's not a moving target. Sorry, it's not a static target. It's a moving target of what the cost is. So I'm not able to give a number. And I didn't fully understand the full list of options, but we are also thinking about different ways, different kinds of vectors that might be useful to even further enhance this platform.
And for both the knockout and knock-in, do you see splice isoform specific effect? For example, depending on whether the guide RNA is targeting one splice isoform, or maybe that exon is gonna not matter anymore in a different splice.
It's a great question, and it's just not something that we've done a lot of analysis or experiments targeting, but I'm sure that there are those kinds of effects, and it would be very, very interesting to look at.
Okay.
We do tend to bias. I think we're using a lot of off-the-shelf libraries, which often go after the first exon to try to maximize the chance of gene disruption and avoid. But whether there's other libraries that are worth thinking about that would go after different exons and see if we see differential effects. It's certainly an interesting question.
And have you assessed the potential off targets for this CRISPR experiments?
A little bit. Where it's as we move towards clinical applications, it's definitely one of the things that we'll need to do to go through the regulatory process. It's certainly something that needs to be considered carefully. There are new Cas9s that have lead to lower off targets. This high affinity Cas9, for example, and or high fidelity, and I think that will help to reduce some of the off targets. It does need to be looked at carefully.
So for the perturb-seq or crop-seq experiment, it's really nice to see the cell population shift in terms of the differential gene expression program. Do you find this perturb-seq to be like a robust result to get you the differential gene signatures? You didn't actually go over that in the talk.
Yeah, it's a good question. I mean, I just, you know, the current data is still relatively sparse, and so picking out the full gene programs is still difficult from the perturbed seed. You know, I think we see differences in state more clearly, and fully getting to the gene programs may require going back and doing some more sort of targeted bulk sequencing once you have individuals. So I don't have a perfect answer. We're still figuring out the right mix of improved analysis and improved techniques to get more fully fleshed into those transcriptome-wide effects.
All right. So I think that's all for today. Our time is up. Thank you so much for that really wonderful talk. It's really exciting. I mean, I think CRISPR has so much potential, and we're just seeing the beginning of its impact. And I look forward to a lot of new exciting work from your group. Thank you so much.
Thank you so much for having me. It was a pleasure.
All right.